So here's our video on cardiac muscle and conduction system. We'll break these out into 5-minute segments for you. So here we go. We're going to talk about muscle histology first and do a brief overview. You should have covered cardiac muscle histology in Biology 201. So if you're weak or not remembering, please go back and review your notes and or text regarding cardiac muscles. These are branching fibers. They are striated, so just like skeletal muscles, they are striated. These guys have one nucleus, again, same as skeletal muscle, but in the case of cardiac muscle, that nucleus is in the center of the cell. In skeletal muscle, it was pushed off to the side. So some differences just structurally and histologically between skeletal and cardiac muscles. Again, these fibers are branching, and they connect end to end. But as opposed to nice, even regular ends, we have a lot of irregular transverse ends. So what happens is we have a thickening of the sarcolemma near the end of the fiber. It does run transversely, it is thicker, and that connection is called an intercalated disc. Intercalated discs contain two different types of cell junction. One type is going to actually hold those fibers together. Those are the desmosomes. If you don't remember a desmosome from Bio 181 or even Bio 201, please go back and review that, but that's going to be the anchoring junction that's going to hold those fibers together. The other type of connection we're going to have between these fibers are called gap junctions. Gap junctions are going to be important to the cardiac fibers because they are what's going to allow the muscle action potentials to transmit from one fiber to a neighboring fiber, and that's going to be key in the actual contraction of both the atria and the ventricles. Remember that there are going to be a lot of mitochondria in these cardiac cells, and that's because we have such a great need for ATP production here. So this is a publisher drawing of a cardiac myofibril. Remember that a myofibril is a muscle fiber. If you don't remember any of these labeled parts, if you don't remember the significance of Z lines versus M lines, if you don't remember an I band versus an A band, or what a sarcomere is or a sarcolemma, the importance of the sarcoplasmic reticulum, please go back into your text or online, find a good review for sliding filament mechanism. That mechanism applies. So all of the depolarization, repolarization, hyperpolarization terminology that you've learned previously is important. We're not going to take time to cover it here, but you do need to understand it in order to comprehend what we're about to talk or to go through next. So during embryonic development, a very small percentage, about 1% of the muscle cells that are in the heart form a specialized pathway. That pathway is called the cardiac conduction system. This system is made up of a special group of cells. Those cells are called myocytes, and they have a very unique or unusual ability, something that other muscle cells don't have, and that is that they can spontaneously depolarize. They have the ability to generate an action potential spontaneously, and that's vital to what happens here in the heart. So collectively, we call these myocytes, these specialized fibers, collectively we call these fibers the autorhythmic fibers. We call them auto, self, rhythmic, beat. We call them autorhythmic fibers because they can generate their own action potential, and they can do so without any sort of input from the central nervous system to generate that action potential. In that way, they are considered to be self-excitable. They will repeatedly generate action potentials, and in so doing, they will repeatedly trigger contractions. These autorhythmic fibers are then going to be considered the pacemaker of the heart. They're going to set the rhythm for the entire contraction cycle within the heart, and they will actually continue to do so even after the heart is removed from the body. So that's how transplants are able to to work so successfully, and that these autorhythmic fibers will keep those muscles contracting, which is going to keep blood moving, which is going to keep those fibers alive, at least for a short time. So it is the autorhythmic fibers that are going to form this cardiac conduction system. Singularly, those fibers are called myocytes. Collectively, they're called autorhythmic fibers. And overall, they are the component of the cardiac conduction system. So what that means is we have to talk about where they're localized, where they're congregated. And we talk about those in specifically two nodes. So the first node that we talk about is the SA node, or the sinoatrial node. The sinoatrial node is considered the pacemaker. It is the pace setter. It is the pace car for the heart. It spontaneously fires at roughly 90 to 100 times per minute. It transmits its action potentials to the AV, or the atrioventricular node. The SA node located in the right atrium, the AV node located at the junction between the right atrium and the right ventricle. In itself, the AV node will fire 40 to 50 times per minute. So if the SA node fails in some way, the AV node will pick up and fire at 40 to 50 beats per minute. In the event that both the sinoatrial and atrioventricular node fail, which is not a good thing, but if they are suppressed, if there's an electrical conduction issue, if they fail in their ability to spontaneously depolarize, then the ventricular fibers will fire all by themselves. That's a beautiful thing. The downside is they only fired about 20 to 40 times per minute. So not ideal for normal daily activity. If your resting heart rate is somewhere in the 60s to 80s, running at half to a third of that is really not ideal, but they will at least keep an individual going. I'm going to stop this video here at just under 7.5 minutes, and we'll pick up on the actual conduction pathway next.